Hi, I'm Russ Altman from Stanford University, and today we're going to talk about leveraging PharmGKB, the pharmacogenomics knowledge base, for regulatory decision making. First of all, I want to start with a brief disclaimer and thanks. Part of this presentation contains slides that were given to me by Dr. David Litwack from the FDA at the Office for In Vitro Diagnostics. I'm very grateful for him sharing those. And also, just to be very clear, the FDA slides, which represent FDA content, show the FDA logo and are mostly blue. And slides that I have created usually have the red banner, such as in this slide, with the UCSF-Stanford CERSI title at the bottom. The goals for this lecture are first to define the issue of regulatory use of external scientific databases as valid scientific evidence, which is an official term used by the FDA. In order to do that, I'm going to introduce the PharmGKB resource, which we've been building here at Stanford since the year 2000. And PharmGKB is devoted to understanding the impact of human genetic variation on drug response phenotypes. Finally, we want to understand the issues faced by the FDA and regulatory scientists in validating databases that are going to be used for regulatory use. So this lecture really stems from one of the challenges that's highlighted in this paper that was published in early 2016 called A Research Roadmap for Next Generation Sequencing Informatics. This was a paper that brought together FDA scientists, academic scientists, and industry scientists who thought about what do we need to know in order to advance next generation sequencing and its use in the clinic. There were 9 challenges, and I've discussed those 9 challenges in another lecture. And this lecture really focuses on challenge number 8, developing methods for using non-FDA databases for regulatory decision making. In another lecture that I hope you've seen, we discussed this issue, and I pointed out that the FDA can't possibly maintain all the genetic information that is needed to make decisions about next generation sequencing tests. We need to understand which tests are clinically relevant, which variants are important to measure and have predictive power about whether a patient will get a disease or whether a certain treatment will work. And it's just impossible for the FDA to manage all of that data internally. Therefore, they have to respond and rely on external databases. If they're going to do that, how can these databases be evaluated and qualified for use? And what are the rules by which FDA can say this database is okay? In that lecture, I mentioned some databases, including PharmGKB, which we'll focus on. This is simply an example. It's my favorite database, but it's also an example of a database that might be qualified someday by the FDA. The issues you have to think about are where does the data come from? How good is it? What are the standard operating procedures used by the database creators to ensure quality? FDA needs to declare what databases it uses. The companies that submit applications would like to check these databases beforehand to make sure that they're saying things that are consistent with current best knowledge. So that's an important factor. And then, of course, we have to be able to always focus on the source of information about why we're interrogating a certain piece of DNA, what type of variation in that DNA is relevant, and if you can measure all of that, what are you going to use it for clinically? And then, of course, there's this issue that databases are not static. And so how does the FDA look at a database that's going to be updated on a regular basis and make sure that they always have the best data, but that it also has been fully vetted and quality controlled? So this is the home page for PharmGKB. As I said, it's the pharmacogenomics knowledge base. It's been in existence since 2000. You can't see this very well, but it relies on this pyramid of evidence where at the bottom of the pyramid is the primary literature, papers written by scientists about associations between genetic variables and drug response. Drug response has influences from genetics in 2 ways. The system of metabolizing a drug, the way it's absorbed, distributed, metabolized, and excreted, that may have genetic variation that changes the blood levels that are achieved in one patient versus another. There's also the way that the drug works, or the pharmacodynamics. The pharmacodynamics may have genes that have variation in them, and the variation in those genes may affect how well the drug works. So what we do in PharmGKB is we read the literature, we aggregate it into databases, we create individual interpretations of single papers, but then we aggregate those single papers into knowledge. We call them clinical annotations that are the clinical significance of all of these research reports. And I'll give you a flavor for that in a moment. The pinnacle, so to speak, of the PharmGKB process is the creation of a clinical guideline that says if you want to use genetics to prescribe this drug, here's how to do it. And that's something that is written specifically for providers, and I'll show an example of that as well. So we've been working on this, and we hope that it's useful to scientists everywhere, and we certainly hope that it will be useful to the FDA in making decisions about pharmacogenomic tests, which would be an example, perhaps, of a next-gen sequencing test. So how could PharmGKB be used by regulators? This is just a small list of what's probably tens or hundreds of ways. They might be checking for evidence that a specific gene is involved in the response to a drug, because they may be looking at that drug, or they may be looking at a sibling drug, and so they'd like to know what's known about this class. They may be checking for evidence for which drugs are influenced by a certain gene. They may be checking the pathway of metabolism or of action for a single drug or a class of drugs to see what genes are in that pathway and should we be worrying about genetic variation in those genes. They might want to know what haplotypes have been observed for a gene. A haplotype is a series of variations that travel together to create a version of a gene, if you will. And so there might be version 1, version 2, version 3, and it may be that certain versions are associated with a drug working, other versions may be associated with a drug not working. And then they might be doing all of this to get a sense for what drug-drug interactions might occur and might even be exacerbated because of the genetic background of a patient, whether there's safety issues for certain patients with certain genetic variations, and whether there's efficacy issues. Will the drug even work based on some issues in genetics? So these and many other uses might be relevant to the FDA mission, and they're all questions that could be answered with PharmGKB. So let me just give you a little sense for PharmGKB. And in almost all these slides, it may be difficult for you to read them. The PharmGKB is a publicly available resource, and I would encourage you to go to www.pharmgkb.org, and you can find all these pages in a way that will be readable. This is the PharmGKB page for a very famous pharmacogenetic gene called cytochrome P450 3A4, or CYP3A4. And there's tabs. There's a tab with clinical information, research information, an overview. VIP is a very important pharmacogene, so we've identified it as an important gene, and we have a summary. We have haplotypes, pathways, link outs, bunch of stuff. This here, what we're looking at is the set of labels of drugs that are metabolized by CYP3A4. There's some labels in the FDA, the US agency. There are also some labels from the EMA, which is the European equivalent of the FDA. And so a user can come here and see that 3A4 is specifically mentioned in certain drug labels. A very useful feature that our users tell us they use a lot, and we expect that an FDA scientist or EMA scientist might also be interested in. When you dive down a little bit into the CYP3A4 page, one of the things you find are these clinical annotations. And as I said, clinical annotations take several papers in the literature and combine them to make a general statement. So for example, this clinical annotation, which I know you can't read or is difficult to read, refers to the role of CYP3A4 in dosing of the drug tacrolimus. And what it says is there's a certain SNP in the genome that if you have 2 copies of the C allele, you actually may need a higher dose of tacrolimus compared to the average population. Whereas if you have a TT, that is mom and dad gave you Ts instead of Cs, then you might be a lower dose. And if you have 1 C and 1 T, one from mom, one from dad, you might need kind of an average dose, or in this case, a slightly increased dose. So that's one summary of several papers that have been read by the curators. PharmGKB is all done by manual curation. We write algorithms, but at the end of the day, things are only put in the database because a human decides to put them in the database. And one thing I'd like to draw your attention to is this light blue section, which provides a level of evidence. And in this case, it says 2A, and there's a little question mark where somebody says, well, what does 2A mean? Could find out what 2A means. And I'll show this to you in a second, but basically what 2A means is that it's not the highest level. There's a couple of papers, but this is not what we would call level 1 evidence, which is the best pharmacogenomic evidence. And then there's more clinical annotations below. So in this next slide, this shows the evidence levels that are supported by PharmGKB. And there's 1A, 1B, 2A, 2B, 3, and 4. And then there's textual summaries available to everybody about what that means. So level 1A means that there's been a guideline written, or there's been a medical society that has endorsed this association, or it's actually being implemented in a major healthcare system. So these are, if you will, pharmacogenomics' greatest hits, the strongest evidence. 1B is one where there is very strong evidence, and it could have been a guideline, or maybe someday it will be, but is not yet. So it's still very strong, but has not yet made it into clinical practice. 2A means, and that's the one we saw a moment ago, annotation qualifies, it's a very important gene, and there's a lot of evidence for its importance. And I'm not going to go through these. They're available on the site, but I'll just say that level 4 means a case report. So it's an intriguing observation. We have no statistical basis to believe it, but we do record it, because if you get 5, 10, 30 case reports, it might actually mean something. So suffice it to say that we have these levels, and we label all pharmacogenomic evidence in PharmGKB with these levels, which our users like, because then they can decide which level is of interest to them, and they can say, give me all variant annotations at level 2B or above. And that's a very useful query. Sometimes they'll say, give me level 4 or below, which means I'm looking for the things that are not proven, because I want to write a grant to prove that they're true and to raise the level of evidence for that association. So it's been a very valuable and useful rubric for us. PharmGKB also has haplotypes. So 3A4 does have these different versions. There's version 1, 1A, 1B. And this is just a summary slide. You can drill down and press on these and get the detailed definition of which variants at which positions define the haplotypes. And haplotypes can also be associated with levels of evidence and clinical annotations, as shown here. Now I just want to do a deeper dive. I'm going to go back one slide. You can see at the very bottom it says view evidence. So I told you that clinical annotations are based on multiple papers that are then aggregated. Well, let's say you're a skeptic and you say, I don't really know how the PharmGKB came up with this summary. I'd like to see the evidence. If you press that, you get a long screen full of several summaries of papers. So this is now at the paper level. There's a PubMed ID, and it says this, for example, was a study of 52 people from Tunisia. There was a certain p-value to the study, and there's a summary of what the study found. And so you can then go ahead and read every summary of every paper that contributed to that clinical annotation, and you can decide, is the annotation created by the PharmGKB team a fair representation of the evidence that was published? So again, we try to be absolutely transparent about not only what our clinical summary is, but where we got that information from, complete with links to the paper. So if you don't even like this, you can just go read the papers yourself. Part of our effort to be very transparent, and that'll become relevant when we talk about what might the FDA require if they're going to decide to use this in their decision making. This is the research page, which lists variants that may not have yet arisen to the level of clinical aggregation. So I'm just going to jump down to the bottom of the page where you can see every row here is an entry for a variant in the genome that has been associated with that gene. Some of them have been turned into clinical annotations. Those are the ones with the blue, CA, clinical annotations. Some of them are part of VIP gene summaries, very important pharmacogenes, and some are simply purple variant annotations that have never yet been aggregated into a piece of clinical aggregated annotation. So again, in one kind of look, you can see what's understood and what has been summarized into clinical actionable information, and what is still a research finding that has not yet percolated up to a clinical action. The other thing I want to mention briefly about PharmGKB is pathways. You can hardly see it, but there are little tiny, on the far left, there are little tiny pictures of pathways. These are very popular features of this database because instead of focusing on a single gene, they talk about the set of genes that work together to either metabolize a drug or create its drug action, its mechanism of action. In the next slide, we have a pathway for the drug abacavir. Abacavir is an HIV drug. It's a very effective drug, but there is an allele in HLA, the gene HLA, that can cause very severe skin rash, even to the point of desquamation or loss of the skin, sometimes fatal. Those alleles are very rare in European populations, and so abacavir can often be used with very little worry in a population of European descent patients. However, in Asian descent patients, almost 20% of those patients might have the dangerous genetic variant that makes abacavir a very dangerous drug. So, as you can imagine, it's either not used in Asian countries or there's strict rules for testing before use of this drug. Well, what you see here is how the drug actually works. So I'm not going to go into details, but the purple boxes are either drugs or they're metabolites. So abacavir is all over this diagram. The little blue eggs are genes that either are affected by abacavir or metabolize it. And then there are other cellular components here showing the transportation and movement of abacavir through the body. These are very popular features of PharmGKB, as I said, because they integrate multiple genes and show how multiple genes affect the response to one drug. Those list of genes might be useful to an FDA regulator who's trying to decide where should they focus their attention in terms of looking at population variation and where it might affect a gene of interest or a drug of interest. In fact, this page is simply a tabular summary of that table, showing at the top all the genes that appeared in that graphic, and then it shows all the drugs or metabolites that appeared in that graphic and some of the details of the connections in the graph, and finally always ending up with PubMed IDs showing the basic source of information which the curators at PharmGKB used to create that pathway. So I think you get the idea that everything we put up on PharmGKB, we allow drill down to the core published literature, which is the basis for everything. And we try to be transparent about it, and our real challenge is presenting things simple to people who want a simple answer, but then allowing them to drill into the complexities if they're about to make a big investment of resources or time into that fact, and they really want to make sure that the fact that we're presenting is true. And as I said, these often end up as published guidelines for practitioners that say, if you want to use the drug abacavir and you have access to HLA variations, here's how you can safely dose abacavir using the information about the HLA variants to protect your patient and make sure you optimize outcomes. So the CPIC Consortium, Clinical Pharmacogenetics Implementation Consortium, CPIC, publishes these on a regular basis, and they can form the basis for a practice of pharmacogenomics. And this is just one page on PharmGKB where we've actually automated the use of the guidelines so that if you type in or select from a pull-down menu the variants that are found in a patient, the website will actually say what the clinical advice from that guideline is. So in this case, the answer is abacavir is not recommended and should be considered only under exceptional circumstances because the patient has one copy of that dangerous allele I was talking about before. Okay. So I want to say a little bit about standard operating procedures. If PharmGKB were to be adopted by the FDA as a credential database, and by the way, as of today, May 2016, it has not, and we have not applied and we have not had discussions about it, but we are thinking about it. One of the things we're pretty confident the FDA would care about is our operating procedures. How do we onboard our staff of professional curators, usually masters and PhD level scientists who do all of the work that I've been showing you? And what do I mean by standard operating procedures? It's a primary way to judge the quality of a database. They should be written procedures They should be explicit, handling edge cases or giving guidance to the curator about how to handle an edge case. They should be available to inspection by all users and certainly by regulators. Hopefully, they reduce the variation in human curation by having people be more on the same page, literally, in terms of how they do these activities. They allow us to maintain at least minimal quality controls, and they establish transparent criteria for data quality. Of course, at the end of the day, we're counting on our professional curators to abide by these procedures, and we also have procedures for checking that people are obeying the procedures. And you can do that ad infinitum. So these are all very hard to read slides. Contact me if you'd like to see the details, because these are not publicly available, although they're not secret. Most people are not interested in looking at our procedures. This is an internal intranet that our curators use. This is the table of contents for the curation standard operating procedures. There are topics like annotating variants, and its clinical annotations, VIP, those very important pharmacogene annotations, pathway, etc. How to edit the website, how to do other curation activities, tools and resources in the outside world that you can use to support your work. Every new curator who's brought onto the team goes through a boot camp where they are exposed to this standard operating procedure, and they're even tested on it with some sample problems to make sure that they've understood how we would like them to do curation. So here's the page on variant annotation. Of course, I'm not going to read it, but it says things like, very detailed, like click on this button, enter an RS number, make sure you've checked for the accuracy of the RS number by cross-checking it to a database called dbSNP, and then goes on and on and on. But for a brand new curator, eventually this becomes second nature. For a brand new curator, this becomes the cookbook for how to create an annotation of a variant. Similarly, we have a page of tips and rules. So there's a difference between the rules, which you must follow, and tips, which are just things that the curators have figured out over time that help be more efficient or effective. Make sure the p-value you are entering matches the phenotype you entered in another field. So it's just little things that can go wrong that are captured and create a kind of a best practices, which is really what the standard operating procedures are all about. Here's a clinical annotation. These start to be very serious. Everything is serious, but these are very serious because they can wind up in patient-related guidelines. And so they say, do this on the web form, filter for the drugs, write a description for each genotype, make sure the sentence should start with a capital letter and end with a period. That's because we do automated quality checking, so we need to recognize the beginning of a sentence and the end of a sentence so that we can pull it out and use it in multiple places. So just every ultimate level of detail, not the most exciting, but exciting to me as the co-PI because it guarantees that the curators are always doing consistent work from year to year and even with a changing staff. And finally, we have a pathway SOP. How do you build a pathway? How do you annotate it? I showed you that table of all the genes and drugs and PubMed IDs. This is how they follow that template. So it becomes very important to think, what would the standards be for the FDA if they were going to evaluate PharmGKB? They have limited resources. I can guarantee them that there's no way they can replicate the PharmGKB. It's taken us 16 years to build it and many millions of dollars, and we have highly trained curators who I would be irritated if they were recruited away to the FDA. And yet we want to make this available to them. So they have to rely on our external database. There may even be some private sector for sale databases that are high quality that they want to use on. How do they decide if a database is appropriate? Hopefully I've made you a fan of the PharmGKB. And what I want to show you now, these are slides from Dr. Litwack at the Office for In Vitro Diagnostics, who has spoken about this issue because it's on their minds. And so this is a very valuable glimpse into how the FDA might be thinking. So the first thing he said is sponsors, and sponsors is the word for people who are submitting a device or a diagnostic for approval. Sponsors must submit evidence to support the claimed intended use of in vitro diagnostics in marketing submissions to the FDA. A marketing submission is the submission which is basically request for approval of a diagnostic. And the whole basis of FDA's ability to regulate these things comes from the Code of Federal Regulations, a large code which has many sections. One of the sections is 21, subsection 860.7, which discusses valid scientific evidence. And it says, and I will read it because it's not that long, FDA relies upon only valid scientific evidence to determine whether there is reasonable assurance that the device is safe and effective. A next-generation diagnostic, I should say, is considered a device. After considering the nature of the device, the commissioner will determine whether the evidence submitted or otherwise available, I have that in italics, actually David has that in italics, because that's the key thing here for a database. They're not submitting our database, but it is otherwise available information to the commissioner is valid scientific evidence. And you can go Google the entire Code of Federal Regulations. So the key thing is or otherwise available. And in fact, this is a red slide, so this is my slide. I actually Googled the Code of Federal Regulations, and there it is, and you can read it, and it's lovely. It's in a fixed font Courier, and it goes on. And it's current as of April 1st, 2015. So it hasn't been modified in a while. Begs the question, what is valid scientific evidence? So best practices at FDA tell them that the valid scientific evidence is from well-controlled investigations, research projects, partially controlled studies, studies and trials perhaps that don't have matched controls, well-documented case histories conducted by qualified experts, and reports of significant human experience with a marketed device. That's important because there may be something approved in Europe which there's a huge body of information about, and FDA wants to be able to use that information to make its decision. And the evidence may vary according to the characteristics of the device, how it's going to be used, what kind of warnings and restrictions are anticipated, and previous experience. Now, what about databases? Oh, by the way, what is not scientific valid evidence? Isolated case reports, that's what we would call level 4 in PharmGKB, we agree. Random experience, don't even know what that means. Reports lacking sufficient detail, and unsubstantiated opinions, and there's plenty of those. So for databases, this is an example where the evidence is generated by multiple parties. In our case, it's the curators and the papers that are written by the scientific community that they read. So the test developer would not be required to be the entity that generates the data. The diagnostic company won't have to create the PharmGKB, but they may want to use it. Genetics and genomics could be served well by community efforts to aggregate and evaluate data, is what David says, the FDA employee. And he lists some of my favorite databases, including PharmGKB, as databases that are of interest to the FDA. And he points out, as I think I've made this point, that this could alleviate the need for each test developer to independently gather the evidence that a certain variant is associated with a certain clinical outcome, which would be part of their claim when they submitted the device. So there are some principles that FDA has recognized. What assurances would they need? They would want it to be transparent. That's why we were thinking about transparency as we built PharmGKB. Well, it's not why. I should say, he's independently come up with these, FDA has independently come up with these criteria, but it's fun for us to look at these and think, how well are we doing based on our own calculations about what would be the best way to build PharmGKB. So transparency, version control, I didn't mention that, but we do updates, and there is the issue of making sure that the update is a quality update. The data quality, the documentation, I've discussed standard operating procedures. Do we have the appropriate expertise? Who do we hire? What's their resume look like? And do they understand pharmacogenomics? And conflicts of interest. Is there any reason why we would put a spin on the information in pharmacogenetic in the PharmGKB for reasons having to do with financial or intellectual property that might taint or otherwise make someone hesitate to trust the information in the database? And so he was, in his talk, David was wondering, could he draw a parallel between the legal definition of valid scientific evidence and how evidence curation and interpretation is conducted in, for example, ClinGen, ClinVar, PharmGKB. So I think he's thinking along the same lines that we were independently thinking just because we wanted to generate a high-quality database. He discusses an example of the Myriad BRCA analysis, where FDA reviewed Myriad's procedure for variant interpretation, and as part of their submission, Myriad conducted a study, and they made a deal that the novel variants identified by this test will be provided to the FDA in Myriad's annual report. So I include this slide because it's an example of a private database that the FDA is going to be using. Once the FDA has it, they can't disclose it willy-nilly to other companies, but they can certainly use this information to consider other diagnostic tests that are submitted and whether they want to approve them because they have or have not been able to detect these kinds of variants. Okay, so that is a summary both of PharmGKB and of current FDA thinking, as well as we can understand it, about the regulation and qualification of genetic databases. Outstanding issues include which databases like PharmGKB will FDA actually decide to validate as valid scientific evidence and thus be used as sources of information when they're evaluating new products. I assume they would then tell the industry so the industry could look at those same databases to make sure everything was consistent. Will there be a process for a database to apply for qualification? And then what will be the ongoing need to maintain this qualification as a source of valid scientific evidence? And if there is going to be a process, what will be the scope? Will they look at standard operating procedures? Will they look at all of my staff's resumes? Will they want to look at previous versions of the PharmGKB over time? How many copies and how far back do we have to keep it? Are they going to ask for a copy of the database to archive just for historical purposes? How are they going to manage conflict of interest? And how are they going to validate that the quality is, at the end of the day, high? Great issues, but an exciting time where if we can solve these problems, FDA will be able to leverage lots of public and private research in the area of high-quality genetic databases without having to reinvent the wheel. So to conclude, PharmGKB is an NIH-funded research resource devoted to organizing information about human genetic variation that impacts drug response, pharmacogenomics. PharmGKB has detailed human-curated information about specific variations, their frequencies in different populations, and their phenotypic consequences. As you saw, we allow deep dives into the nature of our data and a lot of transparency through our standard operating procedures. FDA may choose to use PharmGKB as a source of valid scientific evidence in the future. If it does, there'll need to be a process for qualifying the database, and that process, as of today, needs to be formally defined, although the comments on the record from FDA scientists make it clear that they're thinking about this, and I don't think anybody should be surprised if they start approaching some database qualification activities in the next several months or years. So I want to thank you for your attention. This lecture is brought to you by the UCSF-Stanford Center for Excellence in Regulatory Science and Innovation. I'm Russ Altman at Stanford, and I'd like to again thank Dr. Litwick for letting us use his slides, which are publicly available, I believe. Thank you.